So you heard about pI or isoelectric pH of amino acid, right? Isoelectric pH or pI of an amino acid is defined as a pH at which it shows no net charge. Remember, I said no net charge. I didn't say chargeless. I said no net charge. That doesn't mean it has no charge. That means it has charge, but that is satisfied by equal amount of opposite charge. Correct? So the molecule must have equal number of positive and negative charges so that the net charge becomes 0, and due to this property, that molecule cannot remain in solution. It precipitates. So the solubility of the substance becomes least when it has no net charge. Number one. Number two, due to no net charge, it shows no mobility under electrophoretic field or during electrophoresis. So during electrophoresis, it does not move on any direction because it has neither positively charged or negatively charged. So it shows least mobility under electric field and least solubility. But what is most? If a person is least sincere, that means he is a worst student. Similarly, if it is least soluble, then it is most potent what? Buffer. Its buffering capacity is maximum at pI. Correct? The method by which a substance separated by precipitation at pI is called isoelectric precipitation. Correct? The similar principle is used during electrophoresis for separation of certain substances. Correct? Now, if you can recall, in last class, sir might have told you that pI represents mathematically or arithmetically, it is the arithmetic mean of 2 pK, pK1 and pK2. Pi is equal to pK1 plus pK2 divided by 2. That is arithmetic mean of 2 pK. What are 2 pKs? 1 pK for carboxylic group, another pK is for amino group, because amino acid has got 2 groups, but there are many amino acids which has more than 2 groups. So for them, they will have 3 pKs. So the question arises, between which 2 pK should we calculate for pi? Correct? So for that, I have shown you a table for all amino acids with their respective PIs. Up to this, see, no issues. There are 2 pKs. So do the arithmetic mean, you get the PI. Right? What is to be noticed here? Glycine, alanine, valine, see, all around 6. All around 6. Right? All around 6. 5 to 6. Now, the moment one extra amino group or carboxylic group is added, it changes the range. See, it is now 2.7, 3.2, or see. So what is the difference here? If there are more than one carboxylic group, then pi is in acidic range. Aspartic acid, glutamic acid, in acidic range. Now tell me which one is having 2.77. is arithmetic mean of which two? This one and this one, this one, this one, or this one, this one? কোন দুটো? কোন দুটো সংখ্যার মিন 2.77? বল 1.88 আর 3.6. Correct? So third pk is not at all considered. Why? Who decides which pk to be considered and which not to be considered? Who decides? It is experimentally found out. The pi should be the pH at which it has no net charge. You have to find out between two forms. Which two forms? They exist as no net charge or Zwitterian form. Whichever these two pK in between you will find in Zwitterian form, that is the pi. It may be pK1, pK2, it may be pK2, pK3, it may be pK1 and pK3. It depends between which two pKs the Zwitterian form exists. And a simple number. Just remember what is what. Okay? There is no specific rule. You have to find out experimentally. How do you do the experiment? You prepare 1.1 normal HCl solution and dissolve the amino acid. Right? And then go on adding 0.1 normal NaOH 1 ml each time. Add 1 ml of NaOH solution, mix it, measure the pH. Put a dot in the graph. Like that you continue and you get a graph like like this. Okay? So this is the midpoint of pK1, this is the midpoint of pK2, and this is the midpoint of these two called pi. Okay? So at this pH, it is in Zwitterian form. It is in Zwitterian form. Is it important? It is very, very important. Because the pi of individual amino acids dictate the pi of final product. Depending on how many basic amino acid it contains, the pi of protein might be changed, altered. It might be in acidic range, it might be in alkaline range. Similarly, for histidine, see, find out in which two six and pK2 and pK3. So why histidine? What is the group present in histidine? Imidazole. Correct? Imidazole. Now, so this is pi and this is how the experiment is done. Right? And the graph is not drawn from here. It is drawn from this side. Huh? Sorry. Hey, we are adding base, right? If we are adding base, then from this side. Certain amino acids are there which are not soluble in acid. For them, reverse is done. Mostly, the amino acids are soluble in acid solution, 0.1 normal HCl. What is this? Any idea? In last class, sir have taught you regarding few reactions of amino acids. Among them, one reaction he could not finish. That is, write down ninhydrin reaction. This particular test is specific for amino acids, free amino group. Free amino group. Drink water. So ninhydrin molecule, two ninhydrin molecule gets condensed to form this big compound. It has got name called Ruhmans or Ruhmans purple. Ruhmans purple. Right. So this particular color can be measured at 5-7 nanometer to find out the concentration of amino acid. So can we test presence of amino acid in a solution? What is the test? Ninhydrin test. What will happen? We mix ninhydrin solution and then we have to apply heat. How? Slight heat is sufficient. Heat, not boil, just heat it. If the color changes to purple, then it contains any of the 19 amino acids except proline. The proline does not give this reaction. It gives slightly different reaction. So it produces different color. All other amino acids produce purple, whereas proline provides which color? Yellow color. Yellow. Yellow, yellow, proline fellow. Okay? So yellow color is a color for ninhydrin reaction with proline, because proline is not an amino acid. It is an amino acid, secondary amino acid, not primary amino acid. Okay? Okay. So the ninhydrin reaction takes place with the free amino group. Can it react with peptide? Yes. Because peptide too have free amino end, but number of free amino end compared to free amino acid is very, very less. 1 in 100, 1 in 1000. So the color concentration will be also 1000 times or 100 times less. So we cannot perceive. It might produce, but we cannot perceive. So the test results negative. Correct. What is the length of usual polypeptide? 100 amino acids. So if 100 amino acid exists discretely, they will produce huge amount of color with ninhydrin. But whereas if they exist in peptide, they will produce very less than 1/100th of the amount of color. So they will produce almost null. Correct. So these two things you must remember. One, how the PI graph is drawn for each cases, and between which two pK should we consider the PI to exist. Number one, what are the significances of PI? Correct. What are the significances? Number one, highest buffering capacity, lowest solubility, and lowest electrophoretic mobility. Okay, high, low, low. Fine. What next? What is the test for individual amino acid? Ninhydrin test. Can it be quantized? Yes, it can be used for quantitative estimation of amino acids. These things should know. Protein classification. Protein classification means the different types of protein, how they are classified. It is classified based on 4 criteria: shape and size, composition, solubility, function, and lastly, nutritional value. Why did I say 4? Because one of them you cannot remember. That's why I said 4. You at least remember 4 in detail. That will be sufficient. Based on shape and size. Based on shape and size. Shape and size means, see, Some proteins are very long fibers. Some proteins are like a bomb, laddu. So these laddu-like proteins are called globular protein. The ribbon-like proteins are called fibrous protein. So when you call a laddu fiber, is langcha you know? Is langcha a globular protein or a fibrous protein? Definitely, Sitavog is fibrous protein. But is langcha a fibrous protein? And just relate the shape. It is 10:1. Length is to width. If it is more than 10:1, length is to width, then it is called fibrous protein. If that is less than that, we call it. If the langcha belongs to Shaktigor, then it is fiber. If the langcha belongs to Kolkata, then it is globular. Right? Those who are from outside Bengal, they don't understand. Sorry. Okay. So it is a ratio between LW ratio. It is called length versus width. Someone is becoming giddy hearing langcha. Right? Examples: keratin, collagen, right? Elastin. Example of globular protein: right? Quickly, हाथ चलाओ, fast. देखा जे हाथ देख ताते আমাকে দেখছিস কেন ওখানে I know I am very beautiful, very good looking, but please don't look at me, look at the board. है ना দেখা গেছে নেক্সট হ্যাঁ ঠিক আছে কাগজটা সরা কাগজটা আটকে দিচ্ছে based on function how many are there 12 wait can you remember all 12 now you can remember these don't look at the exact spelling might differ a little bit step star back right step star back now see how you can remember Contractile protein, actin, one example, and that A has been taken here. These are bacterial originated protein, but they are found in our body. Gustation. Gustation means কে বলতে পারবে? হ্যাঁ, উল্টো, আরো নিচে, এক আঙ্গুল নিচে, টেস্ট, তাই না? গাস্টেশন। ঠিক আছে, এক্সাম্পল দেওয়া আছে, কি করে মনে রাখবে দেওয়া আছে, বাকি কাজটা তোমাদের। Shall I change? লেখা আছে সবার? শুধু পড়াতে বেশি সময় লাগে না, পড়তে বেশি সময় লাগে, তাই না? আমার তো পরীক্ষা না, আমি পরীক্ষা দেবো না, যারা পরীক্ষা দেবে তারা পড়বো, তাই তো? Any question from here? No question? No question? Contractile protein. Proteins which are found in the contractile section. Actin, all muscles are made up of actin or myosin, be it from beef, pork, goat, fish. Ultimately, we are eating actin and myosin. 7-TM means 7 transmembrane receptor. One of the receptor through which, when I entered, one slide was open regarding beta adrenergic hormone. This particular hormone acts through this receptor, 7-TM receptor, right? Storage proteins are used for storage of substances. So, for example, ferritin. Ferritin stores iron, right? Is there any other storage protein for iron? Is there any other storage protein for iron? Hemosiderin. But hemosiderin is not a good storage protein. Why? Why I didn't mention hemosiderin beside ferritin? Why? Okay, see, you want to decide which one to be a good storage protein, ferritin or hemosiderin. If I elucidate the function, right? Now I give you two bowls where you can store currency notes. লক্ষ্মীর ভান্ডার বাজ করে কারেন্সিও থাকে না, হ্যাঁ, রাইট? না, When you put the currency note inside, it stores well, no doubt. Can you take it out whenever you need? No. Right? So that is hemosiderin. Right? I give you another option. Your father's pocket. You keep money in your father's pocket. You put 100 rupees in your father's pocket. Next day, you will find 200 coming out. That is ferritin, where you can give and take. So ferritin is used for storage of iron, from where iron can be used. Hemosiderin cannot be used. That is gone forever. You have to break. Then only you can get the money. And hemosiderin is such a thing, when you break, you see inside there was some water and all the nodes have become soiled. So that is hemosiderin. Is it okay? Let's explain. Understand? Hemosiderin is something, even if you break it, rupture the hemosiderin molecule, you won't be able to use the iron present inside. Right? That's why hemosiderosis, hemochromatosis are dangerous disease which causes tissue damage. Myoglobin is used for storage of oxygen. Again, who stores oxygen better? One who can give up oxygen easily. Who transport oxygen better? One who does not give up oxygen easily. কি বললাম? ভালো স্টোরেজ সে হবে যে তাড়াতাড়ি অক্সিজেন দিতে পারবে, আর ভালো ট্রান্সপোর্টার সে হবে যে তাড়াতাড়ি দেবে না। যেমন সুইগি, তাই না? সুইগিতে খাবার অর্ডার করেছে, বিরিয়ানি আসছে, পাশের বাড়ির কাকু লক্ষ লক্ষ রাখে, যেই তুমি বিরিয়ানি অর্ডার করো বাইরে বেরিয়ে বলবে হ্যাঁ আমি তো মিস্টার ভৌমিক, আমার নামই আছে। সুইগি দেবে? দেবে না তো। আপনার নাম কি, ফোন নাম্বার, তাই না? তো হি ইজ এ গুড ট্রান্সপোর্টার। যে চাইলে তাকে দিয়ে দিল, ওরকম নয়, তাই না? Why hemoglobin is transporter? We need hemoglobin transporter like hemoglobin because we have to transport oxygen from lungs till কি tip of the toe, জুতো না, জুতোর ভিতরে যে আমার পাটা আছে তার পায়ের আঙুলের ডগা পর্যন্ত ব্লাডটাকে, অক্সিজেনটাকে পৌঁছাতে হবে। তো কে পৌঁছাবে? হিমোগ্লোবিন পৌঁছাবে। হিমোগ্লোবিন যদি পথেই সব দিয়ে দেয় তাহলে ওখানে অক্সিজেন পৌঁছাবে? তাই হিমোগ্লোবিন ইজ গুড ট্রান্সপোর্টার বাট নট মায়োগ্লোবিন। Based on solubility. Now these classification will never be asked in examination. Happy? Sawai খুব happy. So what are these? It comes protamines, albumins, globulins, histones, glutamines, gliadins, and scleroprotein. Solubility. Soluble in water, dilute acid. Soluble in dilute acid. Soluble in weak acid. Soluble in moderate acid solution. Soluble in strong acid or alkali solution. Soluble in strong acid or alkali solution. Soluble in very, very strong acid. So when I say albumins, it is not albumin alone, singular. Albumin represent a group of proteins which has similar structure or functions. What are different types of albumins? যা আবার চেষ্টা কর, ঠিক করে ভাব কি বলছিস। অ্যালবুমিন কত রকমের হয়? ডিম খাও, ডিম, ডিম, egg খাও। Which albumin is present in egg? Ovoalbumin. দুধ খাও, milk. Which albumin is present in milk? Lactalbumin. মানুষের রক্ত খাও, তাতেও অ্যালবুমিন থাকে। হিউম্যান সেরাম অ্যালবুমিন। তিনটে অ্যালবুমিন ডিফারেন্ট। প্রথম দুটো গ্লাইকোপ্রোটিন আর পরেরটা সিম্পল প্রোটিন, ঠিক আছে? তাহলে হিউম্যান সিরাম অ্যালবুমিন একটা সিম্পল প্রোটিন, এগ অ্যালবুমিন গ্লাইকোপ্রোটিন, ঠিক আছে? অ্যালবুমিনস বলা হয়। এছাড়াও there are many proteins which share the similar molecular weight range along with albumin, correct? Like RBP, retinol binding proteins, which transports retinol. Transcobalamin, which transports cobalamin in the blood, right? So these are all transferring, transports iron, right? Globulin, কত রকমের গ্লোবুলিন আছে এবার বল? আলফা, বিটা, গামা। আলফা দুটো ভাগ, বিটা দুটো ভাগ, গামার কোনো ভাগ নেই, তাই তো? Alpha, beta and gamma globulin. Among them, sit down. Alpha and beta globulin are synthesized from liver and gamma globulin synthesized from B cells, B lymphocytes. Histones, these are nucleoproteins. These are nucleoproteins found in the association with nucleic acid. Scleroprotein. Which protein cannot be digested even by the strongest acid? When you eat something from soil, everything you can digest except one protein. Hair. What is the protein present in hair? Keratin. Now, keratin is the answer from advertisement in the movie, between movies. Alpha keratin. Right? We mammals have alpha keratin. Reptiles have beta keratin. Our nails, alpha keratin. If we eat our nail, it will be excreted as it is. We cannot digest nail. We cannot digest hair. So there is a disease, psychological disease, where the patient has a habit of tendency of eating hair. So the hair becomes like a bolus and gets stuck in the stomach. You have to surgically remove it because it cannot be digested. So it goes and stuck in the intestine. So scleroproteins are very difficult to digest. Based on composition, simple, compound and derived. Simple means which has only amino acid and nothing else. Compound means which has amino acids plus something else. If it is carbohydrate, we call it glycoprotein. If it is lipid, we call it lipoprotein. If it is phosphorus, we call it phosphoprotein. If it is sulfur, we call it sulfur protein or sulfatide. If it is colored complex, then we call it chromoprotein. If it is nucleic acid, we call it nucleoprotein. Right? All these are All these are compound proteins. So compound proteins are called prosthetic group. Derived proteins. These are not found naturally. They are not found naturally, but they are produced during the process of digestion. Incomplete digestion of naturally occurring proteins produce derived proteins. Example, no idea. Anyone? Huh? Cell? Amino acid. That's an amino acid. কি বল চশমা পরে আছিস চিন্তা করছিস বল কি বল তুই তুই খুব মাথা নাচ্ছিস তখন থেকে অতিভক্তি শটের লক্ষণ অত মাথা নালে খুব মুশকিল আমি ধরবো বারবার তুই ভাবিস মাথা নালে ছেড়ে দেবো বস জিলাটিন নাম শুনেছ হ্যাঁ কোথায় শুনেছিস খাবার দেয় একটা খাবারের নাম কর বাবা আরে এরা তো গ্রাম থেকে এসেছিস তুই শহর থেকে এসেছিস তুই তো খাবারের নামগুলো বলিস জানিস বল জেলি আরেকটা খাবারের নাম বল জেলি মন দেখতে হ্যাঁ চামচে করে নিলে ওটা কাপে হ্যাঁ পুটিং আর একটু ভালো হ্যাঁ হ্যাঁ পুটিং ঠিক আছে হ্যাঁ পুটিং ঠিক আছে চলবে তাই না আচ্ছা আচ্ছা বাসে ট্রামে এই টাকায় টাকায় পাঁচটা এই 10 টাকায় 5টা 10 টাকায় 10টা এরকম লজেন্স বিক্রি হয় না যেই লজেন্স হ্যাঁ ও জিলাটিন থাকে ঠিক আছে দ্যাট কন্টেন্স জিলাটিন So gelatin is produced from collagen. Collagen কোথায় থাকে মাসেলে আচ্ছা আর মাসেলে তো বাইরেটা মাসেলে যে বাইরেটা থাকে সেটাই কোলাজেন থাকে ভিতরে থাকে কি অ্যাক্টিন মায়োসিন মায়োগ্লোবিন এসব থাকে হাড় বোনস কোলাজেন থেকে যে কোলাজেন থেকে জিলেটিন তৈরি হয় সেই কোলাজেনগুলো অ্যানিমাল বোনস থেকে ডিরাইভ করা হয় আর খাবি 2 টাকার লজেন্স যেই লজেন্স it is not vegetarian it is taken out from animal bones if available human bones also who is going to see they need some bones whatever animal it may be they have to digest for 48 hours with acid then scrap out the excess material what is found in the bottom of the liquid it is jelly like gelatin but just to produce jelly lozenges they produce gelatin what do you think are they so human that just to produce lozenge toffee they produce gelatin scientists what is the other use for gelatin so agar is there many things are there there is something called alginic acid that can replace gelatin the ice cream you take that doesn't contain gelatin that is the smoothness comes from alginic acid gelatin is used for explosive that is the reason they produce gelatin not for production of lozenges gelatin stick used for explosion ব্যাকটেরিয়া দিয়ে আমাদের কি হবে দুটো মানুষ বাঁচালে নাম হয় না 10টা মানুষ মারলে নাম হয় Suppose you are in hospital and you are saving every day 50 people as a doctor. Next day morning, do you find your name in the newspaper? See Kasaf, what he did. He killed 50 people, 100 people. His name was there. So if you want to famous, kill people, not heal people. Do not heal, only kill. ठीक है ना? Everyone knows. So, Nobel, what was his discovery? Alexander Nobel. Huh? কি বলছে? Discovery of? Dynamite. Just imagine. That was used in First World War. In the same time, Alexander Fleming discovered antibiotic. Who remembers? In whose name Nobel Prize is there? Alexander Fleming or Alexander Nobel? Alfred Nobel. See? So, better change from now. Don't heal, but sit down. So, there are examples of derived proteins. The derived proteins can be of two types, primary derived proteins and secondary derived proteins. We will see. Example of simple protein, human serum albumin. Right? Write down the examples. Nucleoprotein, histidine or histone? Which one? Histone. What is histidine? Thank you. Mucoprotein. Example? Mucopolysaccharide, core protein, proteoglycan, glycoprotein. Example, blood group antigens. Chromoprotein. Example, chlorophyll, hemoglobin. Why can't hemoglobin only example for transport protein? It is also a chromoprotein. Phosphoprotein. All enzymes who participate in regulation by phosphorylation or dephosphorylation. They undergo phosphorylation or dephosphorylation during regulation of a step. So those proteins are phospholipoproteins. Examples of chylomicron, VLDL, LDL, IDL, HDL, free fatty acid, albumin, complex. কতগুলো বললাম? Chylomicron, count. Chylomicron, then VLDL, very low density lipoprotein, then LDL, low density lipoprotein, then IDL, intermediate density lipoprotein, then HDL, high density lipoprotein, then free fatty acid, albumin complex, then LPA, lipoprotein small a. Metalloproteins, carbonic anhydrase contains zinc. Derived proteins, primary derived proteins and secondary derived proteins. Primary derived protein means denatured or coagulated, but molecular weight is not altered. Denatured or coagulated. যেমন albumin. When you boil albumin, albumin gets denatured. So it is no more albumin anymore. It is coagulated albumin, a derived protein. Correct? Collagen to gelatin. Molecular weight is same. Was same with that of the native protein, but they differ in solubility and crystallization. The denaturing agents are heat, X-ray. So multiple X-ray exposure is not good for our health. UV ray, vigorous shaking, acid or alkali. Examples: proteins, myogens, elastin, fibrin. Metaproteins, acid and alkali metaproteins. When proteins are acted upon by acids and alkali, they change their shape along with some addition of hydroxyl ion. They are called acid or alkali metaproteins, not the same like native protein, but similar to native protein structure. That's why metaprotein. Coagulated protein. See, boiled egg, cooked meat. These are all coagulated protein. So what protein do we take? We do not take albumin, elastin, collagen. What we take? The derived protein, primary derived protein. Now, which one is better, to have primary derived protein or secondary derived protein? Which one is better for health? Obviously, secondary derived protein, because it requires less energy to digest. When primary derived proteins are broken down into several pieces, they produce several secondary derived proteins. These secondary derived proteins are having lower molecular weight compared to the primary derived proteins or the native protein. Correct? Few such examples are the intermediates of protein digestive pathway, that is proteases, lower peptide, tripeptide, dipeptide, etc. This is the sequence. Protein enters into stomach, acted on by acid, becomes metaprotein, then it is broken down by the first initial proteolytic enzymes to proteases, then it is broken into further smaller sections called peptones, then it is further broken down into lower peptides, then they are broken down into amino acids. In this region, enzymes are tripeptidase, dipeptidase, aminopeptidase, carboxypeptidase. In this, there are trypsin, chymotrypsin, elastase, collagenase, right? And here renin and pepsin. Renin and pepsin. Here HCl, nothing but HCl, no enzyme. Right? So, পড়া না? পড়া তো? Digestion of protein based on nutritional value. First class and second class. Class 1 and class 2. First class means the protein which contains all the essential amino acids in required amount. Good morning ball. তুমি গুড মর্নিং বস, দাঁড়া না, হ্যাঁ দাঁড়িয়ে লেখ, কেন বলছি পরে, First class protein which contains all essential amino acids in required amount. What are essential amino acids? Which cannot be synthesized in our body. Should be taken from the diet only. Second class proteins which lacks one or more those essential amino acids. Example, animal proteins, plant proteins. Okay? Animal proteins and plant proteins. Proteins like dal, pulses, soybean, proteins like egg, meat, right?